Hi everyone, today I'm going to talk about herpes simplex virus, aka HSV. HSV is a very common virus that infects in human, and there are two main types. HS1 usually causes cold sore around the mouth, and HSV2 is more commonly connected to genital herpes. But actually, both of them can infect either area. HSV is primarily transmitted through direct contact with infected saliva, skin, mucosal surfaces, or bodily fluids. HSV1 is more commonly spread through oral contact, such as kissing. Now let's look at the structure. HSV is an enveloped double-stranded DNA virus. Its structure has four main parts: the envelope, tegument, capsid, and DNA genome. The envelope is the outer lipid membrane surrounding the virus. Embedded in the envelope are glycoproteins that helps SSV attach to and enter the host cells. Because SSV has an envelope, it is sensitive to heat, detergents, alcohol, and drying. If the envelope is damaged, the virus can no longer infect the cells. Under the envelope is the tegument, which is the protein layer between the envelope and the capsid, the brown area that you can see. The tegument contains viral protein that help virus quickly take control of the host cell after infection. One important tegument protein is VP16, which activates viral gene expression early during infection. Inside the tegument is the capsid, which has an icosahedral shape and protects viral DNA. The genome itself consists of linear double-stranded DNA that contain genes needed for replication, structural protein, and latency. HSV belongs to the herpes viridae family. Some related viruses are varicella zoster virus, which causes chickenpox and shingles, cytomegalovirus, and Epstein-Barr virus. These viruses are related because they live in the same family. Unlike bacteria, though, HSV does not have its own metabolism. It cannot make energy or protein by itself. Instead, it completely depends on the host cell. After HSV enters the cell, it sends its DNA to the nucleus and uses the host cell's ribosomes, enzymes, and energy to make a new virus. It shows that HSV can even change the host cell's metabolism to make more energy for viral replication. HSV mainly infected epithelial cells that covers our body and organs, plus our nerve cells. After first infection, the virus can travel into sensory neurons and stay hidden inside ganglia. The viral DNA remains inside a nucleus in an inactive state, the latency. They are sleeping. During those time, the virus produces a few protein, which helps it avoid detection by the immune system. Later on, stress, sickness, UV or a weak immune system can reactivate the virus. This is why some people can get repeated cold sores over and over. HSV also interact with other microorganisms. For example, the gut dysbiosis. HSV1 infections and the antiviral medication used to treat it can cause shift and disrupt the balance of your gut biome. And here are some diseases caused by HSV include cold sores, genital herpes, eye infections called keratitis, encephalitis, the inflammation of the brain, and neonatal herpes in babies. This is why it's important to understand viruses and how they spread. Viruses like HSV are everywhere around us, and we cannot really see them. Learning about transmission, latency, and infection helps us become more aware of how easily viruses can spread in everyday life. And by understanding these microorganisms, we can make a safer choice, protect ourselves and others, and better understand the importance of hygiene health, and disease prevention. Thank you for your time, guys.
Hello everyone, this is my presentation on the hepatitis B virus. The hepatitis B virus is an infectious disease that can cause acute or chronic hepatitis, and it can also increase the risk of other liver diseases in individuals. This virus can be transmitted through direct contact, through the blood, saliva, and bodily fluids, and it can also be transmitted by indirect contact if there is sharing of contaminated objects. Hepatitis B can also survive on objects or surfaces for up to 7 days, and during this time, it is still very infectious. Right now, there are currently no cures for hepatitis B infection. However, there are vaccines that help reduce the spread of the virus. And individuals that already have the disease can be treated with antiviral medication. However, with medications that are being used to combat hepatitis B, they have an effect where they prevent the viral replication of the virus, but they don't have any effect on DNA that is already in infected cells. Individuals who have hepatitis B are also at a higher risk to get hepatitis B, which only affects people who already have hepatitis B. This can happen in multiple ways. One way is like a co-infection, which means an individual can contract both hepatitis B and hepatitis D at the same time, or a person that already has hepatitis B can catch hepatitis D at a later time, and this often leads to even a more severe or rapid liver damage. Hepatitis B is an enveloped virus. On its envelope, it also has surface proteins. As you can see here, there are small, medium, and large surface proteins. They help this virus to attach and to enter the host cells. Apart from the envelope, it has a nuclear core that contains the relaxed circular DNA genome. This DNA is partly double-stranded and the other part is stranded. Surrounding this core, there is also an antigen, referred to as E antigen. As a provider is trying to get testing for hepatitis B in an infected individual, if this antigen shows up, it indicates that the viral replication is active in this moment. Other things that could also be found through the blood when doing testing is non-infectious particles. So these particles can be spherical or filamentous, and so some of them only have surface proteins or lipids that they get from the host, and then also subviral particles, which are only the viral envelope in the capsid, but no genome. So these are things that can also be found in the blood, but again, they are non-infectious. So once the hepatitis B virus has entered the body, it will find the hepatocytes through specific liver receptors. Once the viron attaches and enters the hepatocytes, the rcDNA will be converted to a covalently closed circular DNA. This has to be done because the machinery that is inside of the hepatocyte cells cannot actually replicate the rcDNA. After it has been converted, then RNAs are transcribed from the cccDNA, and they are translated into proteins that can help develop the new DNA. Some of these proteins consist of like viral polymerase, mRNA, and pre-genomic RNA. And all these together also begin to form the core. So as the core develops, pre-genomic RNA begins transverse transcription. So it starts to make a DNA strand from an RNA strand instead of the other way around. And so this leads to the development of the new partial double-stranded rcDNA. And as transcription continues, the rcDNA matures, and then the pre-genomic RNA begins to degrade. And as all this happening, changes from outside of the core also begin, and so this starts the development of the envelope around the core. As everything matures, the virus will move towards the cell membrane and then exit from the cell. As there is an increase in the infected cells in the liver, the immune system reacts, and so it begins to target these cells. However, the immune system is very prominent in the liver. This is due to the gut-liver axis. As we know, the gut is a very big place for the microbiome, and the liver receives a lot of the blood that is from the intestines and the GI tract. So the immune system is trying to prevent any potential pathogens that are coming from the gut and can potentially go into the liver. So in an infected individual with hepatitis B, this immune response becomes even greater. This can potentially lead to scarring of the liver and cause cirrhosis or other liver diseases, and eventually can lead to inflammation of the liver. In a more cellular aspect, the hepatitis B virus can also impact mitochondrial functions. As the virus is using up the host cells for its own replication, mitochondrial dysfunction occurs, and this causes oxidative stress. And this stress can lead to the development of radical oxygen species. And so the radical oxygen species can then damage DNA and also contribute to even more hepatitis B infection carcinogenesis. The radical oxygen species can also diffuse out of the cell and then also cause DNA damage to cells that are nearby. And that concludes my presentation on the hepatitis B virus. Thank you for watching.
Hello, my name is Aaron Kelly, and I did my term project on the avian influenza virus with a focus on H5N1. What is the avian influenza virus, you ask? It is an influenza A virus that circulates worldwide in wild birds. Certain strains can infect mammals and occasionally even humans. It's classified by pathogenicity in chickens. There are two types, the highly pathogenic avian influenza virus and the low pathogenic avian influenza virus. It has a high potential for pandemic outbreaks due to its broad host range and its ability to reassort its genome. It is a constant threat to public health. Let's talk history. There have been documented cases in poultry and wild flocks since the late 1800s, as many as 15 outbreaks globally from 1959 to 1995. H5N1 was first isolated in chickens in 1995, and in 1997 the Hong Kong outbreak killed 6 humans. H5N1 caused hundreds of deaths between 2003 and 2018. The recent H5N1 outbreak in the United States was the highly pathogenic avian influenza virus, which has a 90 to 100% mortality rate in poultry. 82 million birds were infected across 48 states from February 2022 to April 2024. It goes from wildlife to poultry farms to cattle herds and eventually to humans. There were 71 cases in humans and 2 deaths since February 2024. The current public health risk is pretty low, but there's a high risk of continual emergence and pandemic potential. The avian influenza virus has a spherical texture. There are 2 primary surface proteins, neuraminidase and hemagglutinin, as well as a few channel proteins. The virion has a lipid bilayer envelope, as well as an inner matrix protein layer. In the center is the viral genome, which consists of 8 segments of negative sense single-stranded RNA. Replication begins when the hemagglutinin glycoprotein attaches to the host cell membrane, then enters the cell via endocytosis. From there, the virus uncoats and genetic material enters the target cell's nucleus, where mRNA synthesis and replication occurs. New copies of the genetic material leaves the nucleus and assembles and prepares to exit via budding. Here are some fun facts about the virus. Its genus is influenza virus A, and its family is Orthomyxiviridae. Individual strains are named after their hemagglutinin and neuraminidase antigens. For example, H5N1 has the hemagglutinin subtype 5 and the neuraminidase subtype 1. Avian influenza viruses have a total of 16 hemagglutinin subtypes and 9 neuraminidase subtypes for a total of more than 130 subtype combinations. Other relevant strains are H7N9, which began circulating in China in 2013, and H5N6, which was identified in 2014 and has greater than 40% mortality rate. Avian influenza viruses evolve quickly due to antigenic drift, which is where an RNA polymerase cannot proofread, which leads to frequent point mutations. So the virus changes slightly from year to year. Also, antigenic shift, which is genetic reassortment, which happens when 2 influenza viruses infect the same cell. They'll exchange genetic material, which leads to quick and significant changes to the virus. Mortality can be 90 to 100% in birds. Birds shed the virus through saliva, mucus, and feces. It's highly contagious among birds. Accidental infection from birds to humans happens via eyes, nose, mouth, or respiratory passages and can lead to mild to severe symptoms in humans. Birds are the primary host, and they're known to infect humans, domestic poultry, swine, horses, dogs, and bats. A 2024 spillover event infected U.S. dairy cattle. In 2023, ecologists noticed infections in South American sea mammals, including endangered Peruvian pelicans and Humboldt penguins. The end.
Hi everyone, this is James Luna presenting on orthoebolavirus xirense, or Oxirense. Oxirense was originally named Zaire ebolavirus and was first isolated and imaged in 1976. So that's what we see in the top left is an illustration of that original image. There are 4 types of orthoebolaviruses that cause illness in humans, but Oxirense has caused the most outbreaks, though the 2026 outbreak has been attributed to a different type of orthoebolavirus that's pathogenic. Orthoebolaviruses are filamentous viruses of variable length and utilize negative sense RNA with 7 viral genes transcribed by RNA polymerase. The incubation period is 2 to 21 days, and the R0, the number of individuals we would expect to contract Ebola virus after being exposed to the virus, really depends on location. The mean is about 1.95, but it's been as high as 10 in some of the more significant outbreaks. And the disease has a dry and a wet phase. And on the right side, there's an illustration of Oxirense in an infected Vero cell 2 days after infection. Orthoebolaviruses are of zoonotic origin, and the most commonly held belief is that the most recent outbreaks were due to human exposure to fruit bats. Once in the body, the surface glycoproteins of orthoebolavirus bind to neutrophils, which limits our initial immune response, and then hijacks those cells to reproduce more Ebola cells, and they also release cytokines, which leads to inflammation. In the signs and symptoms of orthoebolavirus include fever, inflammation, headache, and sore throat, which progresses to vomiting, diarrhea, and multi-organ failure in the absence of treatment. Without treatment, up to 90% of cases of Oxirense are fatal. Treatment usually involves the administration of IV fluids to replenish the fluids lost, and can involve mechanical ventilation and dialysis for people who are experiencing organ failure or really widespread inflammation and coagulation problems. The reason that these are called hemorrhagic fevers is because the surface proteins of Oxirense and the other orthoebolaviruses bind to and allow entry into blood vessels, which are then damaged. These viruses can work synergistically with other microorganisms by weakening the immune system and allowing for other opportunistic infections. So, for example, the presence of Plasmodium in higher concentrations in deceased patients or subjects also contributed to an increased mortality rate with Ebola, so they can work together. And again, the R0 and mortality rate are really variable depending on location and availability of PPE and quality care. Unfortunately, the reporting on Ebola virus has been really politicized and sensational and othering, which probably led to people delaying or misdirecting their attention in historic and present international aid efforts, and it really encourages us as future caregivers to consider how history and our cultural underpinnings contribute to our concept of our patients and disease, and how we can best serve the populations that we're here to help.
Is on the move. His name? Poliovirus. Polio is small. Very small. Just about 27 nanometers in diameter. You could line up 3,000 polios across a single human hair and still have room to spare. But don't let his size fool you. Polio is built like a tiny fortress. His outer shell, called an icosahedral capsid, is made up of 60 identical sets of 4 proteins. VP1, VP2, and VP3 make up the tough outer armor, while VP4 hides inside like a secret latch door. Polio's greatest trick is that he has no outer membrane or lipid coat, which makes him nearly indestructible in acid, bile, and harsh environments. It's exactly what he needs to survive the journey through the human stomach. Polio belongs to the Picornaviridae family, a name that means small RNA virus. His genus is Enterovirus, and his species is Enterovirus C. His genotype, positive single-strand RNA, makes him a Baltimore Group 4 virus. Polio comes in 3 different variations, PV1, PV2, and PV3. 3 serotypes so different that being immune to one won't protect you from the others. Polio's cousin, also notoriously known to cause trouble, all share the same icosahedral blueprint and share a positive single-strand DNA blueprint. Polio spots protein CD155 on an intestinal cell. The protein is the key to unlocking his capsid shell. He latches on, the capsid twists to open, and his RNA slips into the cytoplasm. Polio's RNA contains the IRES, which is a special code that acts like a trap that will trick the cell into following his instructions. The cell's ribosomes start churning out one giant protein, the polyprotein. Viral proteases 2A Pro and 3C Pro chop up the polyprotein into pieces that polio needs. The viral RNA polymerase copies polio's RNA thousands of times. New capsids self-assemble around the fresh RNA strands, and in 6 to 8 hours, the cell explodes and releases hundreds of polios into the world. The gut bacteria are polio's best friends. Researchers found that mice whose gut bacteria have been wiped out with antibiotics were significantly harder to infect, showed minimal intestinal replication, and less likely to become sick. When the gut bacteria was restored, poliovirus came back full force. The secret lies in polio's ability to grab onto the surface molecules of bacteria, specifically lipopolysaccharides and peptidoglycan, stabilizing the capsid against heat in harsh gut conditions. There are 4 different phases after polio's successful attempt to infect the host. In the first 3 to 21 days, there are typically no symptoms, but polio is replicating in the host cells. In the second phase, the host begins exhibiting symptoms. This happens in 24% of cases, and the symptoms eventually will self-resolve. 1 to 2% of the cases progress to the third phase, where polio reaches the central nervous system. A different set of symptoms then arise. Less than 1% of the cases progress to the fourth phase, where polio destroys the anterior horn motoneurons, causing asymmetric flaccid paralysis. When polio wins, there's some catastrophic damage. Polio travels to the CNS system via the bloodstream and targets motoneurons by binding to the surface receptor CD155. Polio takes over the host cell's machinery to mass-produce viral RNA and proteins. At the end of its cycle, it will lead to cell death and cause acute flaccid paralysis, which is muscle weakness and loss of reflex. The death of these neurons trigger local inflammation, and damaged areas are replaced by glial cells, which result in permanent motor unit loss. Polio can survive in sewage, rivers, and waters for weeks. Transmission is almost entirely fecal-oral, and polio exits through feces, contaminates water or food, and is swallowed by his next host. He can shed in the stool for weeks, and sometimes way before symptoms begin. Humans are polio's only natural host. There are no animal reservoirs and no alternate species for polio to hide in. This is polio's biggest weakness. If every human were to become immune, polio would have nowhere to go, and scientists have found a way to use this against him. Public health teams can test sewage for polio RNA and detect an outbreak weeks before an individual starts showing symptoms. Wastewater surveillance is now one of the most powerful tools in efforts to eradicate poliovirus. In 1988, there were 350,000 paralytic cases per year in more than 125 countries. The Polio Global Eradication Initiative launched that year and set out to hunt down poliovirus to extinction. Jonas Salk created a weapon called the IPV, and Albert Sabin also created OPV. Both are vaccines, with mass immunization campaigns and wastewater surveillance, all efforts to drive polio from this world. By 2014, wild poliovirus was reduced by over 99.9%, and only PV1 persists, cornered to Afghanistan and Pakistan. But polio has one last trick up his sleeve, and the CVDPV problem. The vaccine used to eradicate him can also produce a mimic in communities where vaccination rates are low. The end game requires not just vaccination, but universal vaccination everywhere and all at once. Polio's life isn't over, but for the first time in human history, we know how polio's story will end.
I made.
What is it?
This is a respiratory syncytial virus. It affects the upper respiratory tract, sometimes can affect the lower respiratory tract if you're under 12 months or immunocompromised, leading to bronchiolitis or pneumonia.
How does it spread?
Respiratory syncytial virus, RSV from here on out, can spread by via droplets or a sneeze, cough, or any other upper respiratory fluids. RSV belongs to the Paramyxovidae family and the genus of the orthopneumovirus. The closest microbial relative to this little guy is the human metapneumovirus. RSV is an enveloped negative sense single-strand RNA virus, and its virions are highly pleomorphic. Its structure consists of a pleomorphic lipid envelope, often filamentous, not shown here. With these little teeny tiny proteins, glycoprotein G for attachment and entry into the host, and fusion protein F for penetration.
What does it affect?
It affects your lungs, your nose, and your head. All upper respiratory things are like a cold. This one can be very dangerous.
How does it grow?
That is very interesting. This little guy right here attaches to your ciliated epithelial cells inside your upper respiratory tract. And from there, the RSV mechanism increases the production of virions called hyperglycolysis. With hyperglycolysis, the host airway epithelial cells are reprogrammed to increase glucose uptake, generating rapid energy required to produce new virions. RSV activates a cholesterol biosynthesis in hosts, contributing to lipid-rich structures that stabilize protein F and promote cell entry and cell-to-cell fusion, syncytia. Lastly, RSV alters lipid metabolism, leading to increased free fatty acids in the lung tissues, disrupting lung tissue surfactants, and thus contributing to lung injury, and sometimes thought to be a factor in childhood asthma increase.
Can it kill you?
Immunocompromised in very young children, yes. There have been instances of death. One instance was a 63-year-old man who was hospitalized and unfortunately did pass away from the RSV. Babies are often hospitalized as well, with a prevalence of 61.4% from RSV versus all of the other upper respiratory tract infections that there are in the world.
Is there a vaccine?
There is a vaccine. There is a vaccine for adults over 75 or women who are pregnant, and the 32 to 36 week mark, so that it protects the unborn child for the 6 months after birth.
Does it go between humans and animals?
Not usually. There is only one animal that can actually possess RSV, and that is chimpanzees. Although other animals are not fully permissive of RSV, they are only partially, like mice, which is how they do testing for the vaccine, which is done in mice.
Today, I'm taking you on a surprisingly dramatic journey into the world of Coronavirus, the viral family in 2020 that decided to redecorate our entire planet. This is my deep dive into SARS-CoV-2, what it is, how it works, why it spreads so quickly, and how it compares to its spicy feline cousin. SARS-CoV-2 is a member of the Coronavirus family, a group of crown-wearing RNA viruses named for their spike proteins. Before 2020, most human coronaviruses caused mild colds, quietly minding their own business. This one decided to say, however, I'm built different, and launched the biggest microbiology lesson of our lifetime. Today, let's break down its anatomy and its replication. This virus belongs to the beta coronavirus genus, alongside SARS-CoV from 2003, MERS-CoV, and several bat coronaviruses. One especially interesting relative is the feline coronavirus, which usually causes mild digestive problems in cats, but sometimes it mutates into feline infectious peritonitis, or FIP, a severe inflammatory disease driven by infection of immune cells, a much severe disease. So, same family, different hosts, different types of drama. Let's talk architecture. SARS-CoV-2 is roughly spherical and 80 to 120 nanometers. The major components include spike proteins, which bind to the ACE2 receptor, membrane and envelope proteins, which help organize the structure, and nucleocapsid proteins, which wraps around the RNA genome like a protective blanket. The spike protein is the real star of the show. It's responsible for attachment and entry. Membrane proteins help shape new viral particles, and envelope proteins assist in assembly and release. Because the genome is positive sense single-stranded RNA, it can start making proteins immediately as soon as it enters a host cell. There's no warm up needed, it just takes over. Since we know viruses can't grow and reproduce by themselves, SARS-CoV-2 must hijack the host cells. It binds to the ACE2, fuses with the cell membrane, and releases its RNA into the cytoplasm. It basically takes over and says, hey, I work here now, and then uses its host ribosomes to build viral proteins. Replication happens in a few key steps. First, attachment to the ACE2. Next, fusion and RNA release. Then we have translation of viral proteins. Next, DNA replication by viral polymerase. Then we have assembly in the endoplasmic reticulum and the Golgi apparatus. And last, we have exocytosis, where new viruses get shipped out into the body like millions of tiny little Amazon packages. Now let's talk about what happens when the virus enters the human body and how our immune system responds. On the left, you can see the model of the virus that I made. Those red spikes are what our immune system focuses on first. When the virus enters, our innate immune system jumps in immediately. Things like macrophages, dendritic cells, and interferons try to slow it down. Think of it kind of like bouncers at a club trying to keep the chaos to a minimum. But the real power comes from our adaptive immune system. B cells start producing antibodies that specifically recognize those spike proteins. Meanwhile, T cells hunt down the infected cells and destroy them. This tag team response is what clears the infection. And then, better yet, it creates memory cells so your body remembers the virus in the future. Vaccines take this system and helps give it a head start. The COVID-19 mRNA vaccine basically delivered the recipe for the spike protein only, but not the whole virus. Your cells make harmless copies of the spike, your immune system sees it, and your body builds those same long-lasting memory B and T cells, so when the real virus shows up, your immune system is already ready. Variants show up when the virus makes small mistake, so mutations, while it's copying its mRNA. Most mutations don't matter, but some change the shape of the spike protein just enough so that it affects how the antibodies recognize it. That's why we had variants like Delta or Omicron, and that spread more easily and it escaped partial immunity. The virus wasn't becoming a new species, it was just kind of shopping out parts and upgrading its attachment tools. But overall, the body between the immune system and variants is a constant race. The virus mutates and the immune system adapts, and vaccines help us stay ahead of this by several steps. Feline coronavirus, however, spreads mostly through fecal oral route, specifically in multi-cat environments. SARS-CoV-2 spreads through respiratory droplets and aerosols. What they do share is its adaptability. Small mutations can dramatically change disease severity. Feline coronavirus mutates into FIP, which is a perfect example of how the virus can shift gears. SARS-CoV-2 spreads efficiently because people can transmit it before systems even appear. Aerosols makes it easy for the virus to hang out in the air, especially indoors. SARS-CoV-2 has a simple structure, but it's incredibly efficient. Think small genome plus rapid mutation plus high transmission equals global pandemic. Comparing it to the feline coronavirus, it shows how host species shape disease outcomes. Understanding the Coronaviridae family helps us predict future outbreaks in both humans and animals. Thank you for watching. Hopefully, this gave you a clear and engaging look at the SARS-CoV-2's world, as well as the viral family it comes from. This tiny virus changed our healthcare system, our lives, but studying it helps us prepare for whatever nature decides to throw at us next.
Hi, let me introduce you to the rhinovirus. Here is my origami art project as an example of the rhinovirus. The rhinovirus causes the common cold. It causes symptoms like sore throat, runny noses, sneezing, watery eyes, headaches, and body aches. While usually mild, it can be very serious, especially those with respiratory issues and some children and the elderly. The shape of the rhinovirus is similar to a 20-sided die. It's made up of proteins called VP1, VP2, VP3, and 4. Rhinovirus C, a special type of rhinovirus, has unique spikes on its surface that helps it avoid the immune system. Rhinovirus belongs to the enterovirus group in the Picornaviridae family. It's a single-stranded RNA virus with a genome of about 7,200 nucleotides. There are three types: RVA, RVB, and RVC. The RNA helps the virus make the proteins it needs to infect cells. Rhinovirus doesn't have its own metabolism. It relies on the host cell to provide the energy and materials it needs to replicate. The rhinovirus spreads through airborne transmission and droplets. It has an affinity to our mucous membranes. Once inside our bodies, it attaches to cells in the respiratory tract, gets inside by means of penetration, and quickly makes copies of itself. It uses the host cell's DNA to do this by using the lytic cycle, releasing new viruses to infect more cells. This is a fast process, which is why when you are contracted with this virus, you often feel symptoms days later. Rhinovirus often leads to other infections, like bacterial sinusitis and pneumonia. It can also interact with other viruses, just like RSV, making illnesses worse and increasing hospitalization. As mentioned before, rhinovirus spreads through droplets, contacts with infected individuals, and surfaces. It can survive for many hours, making it easy to spread. The immune response to rhinovirus includes inflammation, which can worsen some conditions like asthma. While usually minor, rhinovirus can cause serious respiratory issues. It affects daily life and productivity, leading to economic costs. For example, it can cause laryngitis, which is inflammation of the voice box. I currently have this at the moment. While I was sick for about a few days or so, my voice was raspy, hard to talk, and was very soft-spoken. As said, it affects a lot of our daily life and can be very cumbersome. An interesting note about the rhinovirus is that there are more than 150 strands of rhinovirus, making it difficult for the immune system to recognize and defend against them all. This is why, even if you get the cold vaccine during cold season, you might still catch the case of the rhinovirus. Often dismissed as minor, the rhinovirus can cause significant respiratory illnesses. Continued research and public health awareness are crucial. In the meantime, maintain good hand hygiene and respiratory etiquette to reduce its spread. Inside the origami viruses that I made has a little box that represents the capsid layer of the virus. Normally, it should be in the inner outer layer of the virus. However, I made it into a small box just to represent that it is inside the rhinovirus and that there are little single strand DNA inside the box. I made this little box here inside just to represent all of that. It is really fun. Hope you like my art project. and hope you liked the little short clip of me cursing at the rhinovirus for getting me sick for about 10 days or more.
Hey everyone, in my presentation I'm going to be talking about the prion protein and its conversion into a more aggressive form of the protein. So here we have a normal prion protein. In the image you can see that there are 4 helixes, 2 small helixes and then 2 of the big helixes. The 2 small helixes are most likely helix A's and the big helixes are helix B and C. The C helix is the helix that is connected to the strand with the red tip. You can find these proteins virtually almost everywhere in the human body, but they're more commonly found in the cerebral spinal fluid, which then travels up to the brain, and once a misfolded prion protein occurs, can do a lot of damage. There has actually been studies showing that the misfolded protein can be found in the dermis of the skin, and once found can be used to see if a person will develop a brain degenerative disorder later on in life. And as for the function of the normal prion protein, we really don't know what the function is, but we can hypothesize that their function is either assisting in synapses, cellular adhesion, and cell-to-cell communication amongst other cells within our body. Now, normally these proteins are harmless, but as you can see in the image to the left, there can be an error in the replication process to where it'll create a misfold in the protein and more commonly in the A helix. But to better understand how this can happen, we got to know the metabolism of the prion protein. So the prion protein undergoes synthesis in the endoplasmic reticulum of cells as a precursor protein, which undergoes post-translational modifications, including glycosylation and disulfide bond formation, to acquire its final conformation. And once this is done, after the synthesis, the prion protein is transported to the cell surface and anchored to the plasma membrane via GPI anchor. That's not the actual name of it. You guys can go look it up, but the name is ridiculously long, and yeah, I'm not going to try that. The GPI anchor allows the prion protein to interact with other proteins and cellular structures. And throughout this process, there's so many ways that this protein could go wrong, which doesn't happen all the time, but it can happen where there will be a misfolding in the protein. The misfolding is the misfold is isoform, or the misfolded isoform has a high beta sheet content and a propensity to form aggregates and amyloid plaques. Consequences of a misfolded prion protein. So a misfolded prion protein could lead to several brain degenerative diseases, including but not limited to Alzheimer's, Puru, and Creutzfeldt-Jakob disease, as shown here on the screen. The sample to the right, control sample, is a healthy brain, and this just shows how devastating the misfolded prion protein can be once it enters the cerebral spinal fluid, and the cerebral spinal fluid pumps it up to the brain, and it forms these plaques to where our immune cells just eats away at these plaques, but at the same time are eating away at our brain because plaques are composed of a spongiform cell of our brain. So what the disease prion protein would like to do is it'll attach itself onto a healthy cell, and then what it will do is basically pump a bunch of fluid into the cell, which gives it a spongiform look. This spongiform look is seen as seen as hostile or foreign to our immune cells, which then come by and inflame our brain, and just everything just goes downhill from there. Um, these types of diseases can affect the way we breathe, can affect the way we move, our thought process, everything. It's basically like putting a virus in a computer, and the computer just loses function in each one of its systems one by one. It's what's going on here, but in a biological form. Um, yeah, I just wanted to add on a little bit more from the previous slide. And while I do so, here's an image of a diseased brain or diseased tissue with Kuru and a normal tissue. But I wanted to add on that there are many ways to obtain this misfold in the prion protein, and it could be through genetics, it can be through eating contaminated meat, whether that be uh, meat from an animal or human meat, which is, I guess, also an animal. And it could also happen spontaneously. So it could just one day your prion proteins just decided not to replicate normally, and boom, you have the diseased prion protein. And these diseases that come after a diseased prion protein are incurable. So it's no matter if you do get it and we find it early on, there's not really a whole lot we can do but give people more comfortable life towards the end of life. Um, the longest I've read that a person can live to was 2.5, 3 years. And yeah,